Well, hello everyone, and welcome back to Science in the Age of COVID-19. Today we are super happy to welcome Shane Crotty, a professor at the Jolla Institute for Immunology and also associated with UC San Diego, my future home, whenever it becomes safe to move there. Shane, you may know him from being an internet sensation. He told us that he recorded a video on RNA vaccines that has over 1 million hits already, which is the science equivalent of Gangnam Style. So Shane has done a lot of work over the years on vaccines, their efficacy, their safety. He's done deep dives into follicular helper T cells, and for instance, the activity of the broad spectrum drug ribavirin targeting RNA viruses, which, as I'm sure you're all aware, SARS-CoV-2 is. And today he will fill us in on immune memory and I'm hoping a little bit of the cell types that we haven't heard as much about from the other talks, maybe T cells and neutrophils, but that will be up to him. Okay, Shane, take it away.
Thank you. I didn't realize you're moving out to UCSD. That's great. Sorry for the delay. So thanks for having me. I am, as mentioned, I'm at the Jolla Institute for Immunology. So for those of you who aren't familiar with this, LJI is a nonprofit research institute on the University of California, San Diego campus. So we're essentially a freestanding immunology department, doing infectious disease research, autoimmune research, fundamental immunology, and cancer immunology. And I'm going to talk today about adaptive immune responses in memory to COVID-19. As part of that, I am going to talk you through our working model for how we think the relationships are between immune responses and COVID-19. But I'm going to give you some background information, introductory science first. And really, I'm going to talk about 3 scientific stories that we've worked on the past year. One is immune responses during average cases of COVID-19. Another is immune responses during acute disease and hospitalized disease. And the third is immune memory to this virus. So this is everything I'm going to talk to you about was done by my lab and Alex Sette's lab, a collaboration between the two, which has been a phenomenal collaboration, and I'll explain. And the first study that I'm going to tell you about is this one, which really was highly impactful in the field. Is probably the first major study of T cell and antibody responses together in COVID antigen virus specific T cell and antibody responses together in COVID-19 cases. And so there's the paper, but here's the live action view of the paper. This is Tony Fauci holding it up in Congress, which was a total surprise to us, but was a fantastic surprise. He was using it as part of trying to explain T cells to congressmen, which was something new. Just at the time, I didn't do much Twitter. So like a popular tweet from me would get like 10 likes. But I sent out this photo and just said, maybe I should make business cards that are just that photo. And it got thousands and thousands and thousands of likes, really, as a sign of the respect and admiration that people have for Tony Fauci. But I thought you'd enjoy seeing some of the things that people basically sent back, and one was that people made GIFs for us, and said, think of all the uses. And I think my personal favorite was this woman who replied, I suggest printing it out on a custom blanket and cradle yourself in sweet validation every night, which was pretty great stuff. So why the attention to that paper? So back in March, there was a huge amount of fear and uncertainty about this virus and about this disease. And one big component of that was people wondering, do people make meaningful immune responses? Do people develop immunity to this virus at all? And we felt like the answer was very likely yes, because it is an acute infection that resolves in most people. But obviously, it was important to generate that data, and in a pandemic situation, you want to generate that data as quickly as possible. So fully understanding what kind of immunity is important against COVID-19 is a complicated and challenging and multi-layered question that really might take a number of years to fully answer. But we realized we could get a pretty quick answer, a pretty quick indication about what components of the immune system were likely to play roles by looking at average cases of COVID-19. Because with SARS-CoV-2 infections being an acute infection that resolves in most people, what that probably means, right? What that probably meant was the virus infects people and gets going, and then people make an adaptive immune response that catches up to the virus and clears the infection. And so in that scenario, by asking, okay, in people who have had an average case, and by average, I mean, we mean cold or flu-like symptoms, non-hospitalized. In average cases, people who got sick and then got better, if we now take a look at their immune response after they got better, that can give us indications about what kind of immunity is important against COVID-19, because these people made immune responses that resulted in them getting better. Okay, and so there are three major components of the adaptive immune system, which I think most of you will be familiar with. So there are antibodies, CD4 T cells, and CD8 T cells. From a vaccine perspective, antibodies are important in almost all currently licensed vaccines. CD4 T cells are important for multiple different reasons. One is that in almost all cases, the CD4s are required to get a high quality antibody response, to get a neutralizing antibody response. And those would be the T follicular helper subset of CD4 T cells that we have expertise in. But in addition, CD4 T cells are complicated, and there are a number of different helper and effector functionalities they can have. And in the context, it's certainly long been known that they can have antiviral activities, be that Th1 cells or CTL type cells. But notably
hey Shane.
Yeah.
Hey Shane, we're still just seeing Tony Fauci. Are you
really?
Yeah.
Yeah.
While that's pretty gratuitous, that's not what I'm seeing, just FYI.
Oh,
so yes.
okay. Yeah, maybe
looks like maybe by hitting the back button, something changed. Let me just opt out of it and pop back in. Yeah, it looks like when I hit the actually, so then I should. Okay, so can you see the GIF now, or did it?
I just see Shane has started screen sharing.
All right, let's go ahead and get out of this. I'm only one slide ahead of you, so it won't
okay.
But yes, the goal is not to have an entire seminar of showing.
I mean, he is awesome to look at. But
that's funny. All right, so are we sharing again?
Yep. So we see Tony doing his thing.
The GIF?
Yeah, we see the GIF.
Okay, because it had been on the. So it's just showing the GIF.
Oh yeah. Okay. Yeah. Okay. Good. We didn't even see that before, so I think you're good. Yeah. Okay.
Okay. Yeah, that was only supposed to be up for a moment, but
okay. You're in the money, Matt.
So here's the slide that I was on. So antibodies, CD4s, and CD8s with this summary, and antibodies, and then here I was on the CD4. So CD4s are critical for the majority of antibody responses, but they can have other jobs. Interestingly, in the case of coronavirus infections, it was shown by Stan Perlin's lab back with original SARS, classic SARS, that CD4 T cells could actually protect mice independently of antibodies or other functionalities, indicating a potential protective role in CD4s against respiratory coronavirus infections. And then the CD8s, I mean, classically, they're certainly well known to be important in many viral infections, and they directly kill infected cells. And really the oversimplified view of the immunological universe would be that antibodies are very good at stopping viruses outside of cells. But once you have an active infection and viruses are inside of cells, CD8 T cells eliminate those. And CD4s do some of each, essentially. Okay, so the first study, we enrolled 20 people who had, again, average cases of COVID-19, and asked what did their antibody CD4 T cell and CD8 T cell responses look like. And we really feel like it's important to look at all three of those major branches of the immune system to get a real sense of what types of immune responses may be important in controlling an infection. And so that's challenging with the T cells, because T cells recognize peptide epitopes, and those epitopes can be difficult to predict, particularly for a virus like SARS-2 that has a relatively large genome of 30,000 bases. There are a lot of different potential epitopes there. And luckily, the Alex Setti is one of the best, is one of the best people in the world at predicting T cell epitopes. And so because of that expertise, they started very quickly predicting and trying to identify T cell epitopes, sort of modeled in this little Lego model of SARS-2 here in humans. And we use those peptide pools to measure the T cell responses in people. And what we observed was that 100% of people made antibody responses to spike. And so that was good. That connected our study with other studies that were all serological. But also 100% of people made CD4 T cell responses to the virus, and 70% of people made CD8 T cell responses to the virus. And those were measured with these predicted epitope pools made by the Setti lab. And so that was valuable. And those have now been shared by the Setti lab with, I think, 100 different labs around the world, so that labs in various contexts and countries can measure T cell responses to SARS-2 on their own. And an additional key feature that we were concerned about was that it was clear already that almost all of the vaccine efforts were going to focus on spike, the S. And from an antibody perspective, that certainly made sense, but from a T cell perspective, that could be problematic. And certainly for other viruses, such a protein can sometimes be a rather poor T cell target. So it was important to us to measure T cell responses to spike. And we were happy to see that spike was actually quite a good target of both CD4 and CD8 T cell responses, which was an encouraging sign for vaccine development. That selection of spike as a single antigen for a vaccine was a reasonable selection choice based on these data. Additionally, since this was a new virus, we really wanted to make sure we weren't missing a major component of the T cell response. And so Alex Setti really insisted that we look at every single one of the 25 proteins across SARS-2, which is a challenging set of experiments, but was done. And the key result from that was that the T cell response to the virus is quite broad. So that, again, was a positive sign that the virus was relatively easy for T cells to recognize. And interestingly, we couldn't immediately make sense of the pattern of the T cell responses, and then we reorganized them based on inferred abundance of the viral proteins. from most abundant to least abundant. And sure enough, the CD4 T cell responses largely track with the abundance of the viral protein. That again was a good sign that the virus was largely playing by immunological principles we understand. And notably, spike is a highly expressed protein, and essentially every one of the people was recognized was making T cell responses to spike. Again, a good sign. So overall, the take-home message from that study was good news that at least in these average cases of COVID-19, the antiviral immunity largely matched expectations. That is, for a respiratory viral infection, a novel respiratory viral infection, the antibody response looked reasonable, and that for most people, both the CD4 and CD8 T cell response were engaged. And I'll talk some more about the phenotypes of those later. And additionally, that again, the T cell responses were broad. So from that point, which was a reference point for a lot of subsequent immunological studies as well as vaccine immunological studies, we then moved to look at adaptive immune responses in acute cases of COVID-19. This is much harder to study. Well, first of all, so this is obviously important because one wants to know why do some people end up in the hospital and some people not, right? What is the source of that heterogeneity? And specifically for us, trying to get a sense of, does that have anything to do with adaptive immune responses? And it was clear that among hospitalized patients, people in the ICU, people with severe COVID-19 do have inflammatory signals, right? And so it's been important to try to understand, is that inflammation and inflammatory pathology associated with adaptive immune responses or innate immune responses or some other process? And would that relate, how would that potentially connect to vaccines or treatments or other issues? Okay. These are much harder studies to do because you have to get blood samples from people while they're currently sick, right? And it's also, to understand what's going on, it's important to collect samples from people across a range of disease severities. And additionally, we felt it was also immunologically important to get samples from people relatively early in the course of disease, but then still have be people that we track throughout the course of their disease. And that's because once somebody's in an ICU, an intensive care unit, many things could have gone wrong by that point, and it can be difficult to interpret cause and effect, chicken and egg kind of aspects. Okay, so that's the way we set up this study. We enrolled a total of about 52 individuals, and again, like in our previous study, for us it was very important to be measuring CD4 T cells, CD8 T cells, and antibodies all at the same time in the same people, and doing so in a virus-specific way, directly measuring the virus-specific CD4s, CD8s, and antibodies. So we did that. I'm going to take you through the data pretty quickly and then focus on the summaries of the data, but I'd be happy to talk about technical details, technical aspects of the data in the Q&A. We measured a variety of antibody responses. I'll say most notably the neutralizing antibody responses, and for this we used pseudovirus assays, but those correlated well with live neutralizing antibody assays, live virus neutralizing assays, which lots of labs have observed at this point. And we basically have 25 acute subjects and 25 convalescent subjects, which are separate from the previous study. We then measured T cell responses in all of these people. Here we're looking at the CD4 T cell responses in unexposed, acute, and convalescent individuals. Since this is a new viral infection, we wanted to do the assays in as agnostic a way as possible and not depend on a single cytokine. And so for this, we use what we call activation-induced marker assays, which are cytokine-independent antigen-specific T cell assays. Here we use our preferred combination of surface CD40 ligand and an OX40 for the CD4s, and you can see it gives very nice measurements of the response. And then this is the predicted epitope pool by the SETI lab. And then here we're looking at the totality of epitopes in the totality of spike, M, and nucleocapsid. And then we could take all 4 of those responses and combine them together to give us a combined metric of the CD4 T cell responses in people. Okay. We looked at the functionalities of those CD4 T cells in a number of ways. One way was to look at secreted cytokines. That was a live cell assay, so we could look and see what the cytokines were that were secreted. And they predominantly matched antiviral immunity expectations. Interferon gamma and IL-2 were the primary cytokines measured. There was concern about the potential of Th2 cytokines in people with severe disease. We really see no evidence of that, and most other labs see no evidence of that. And a similar thing with IL-17. We're using CMV as a reference point as what one sees in another viral infection. We also used intracellular cytokine staining, ICS assays, as a more common experimental technique, and we saw similar outcomes there. That again, the primary cytokines observed were interferon gamma and IL-2. Again, overall matching antiviral immunity expectations. Among the CD4s, TFH cells are a subset of those. So these are the cells that are specialized for helping the antibody response. And they can generally be identified by expression of this chemokine receptor CXCR5. So amongst the virus-specific CD4s, TFH cells were a relatively large subset. For the CD8s, we did similar assays and similar approaches, again, with predicted epitopes or overlapping epitopes against spike, M, and nucleocapsid. And we could combine the data here, and we could do functionality assessments of the cells by looking at interferon gamma and granzyme B and intracellular cytokine stains. So if we take all of those data together and other data not shown, we could look at it several different ways. And so the simplest way to look at it was to take antibody responses or CD4 responses or CD8 responses and look for associations between adaptive immune responses in people and disease severity. And we refer to severe disease as essentially, the pretty standard clinical definition among hospitalized patients, and mild would be non-hospitalized patients. We have quantitative metrics of this as well that'll be in later slides. And these are referring to peak disease severity in each person. So with that, we could say, okay, did neutralizing antibodies correlate with disease severity? And the answer was no. And that's largely been seen in much larger studies than ours that did specifically serology. Frankly, it's much harder to do T-cell assays than antibody assays. You have to have live cells. It's more expensive. It's more challenging. It's probably like 10 times harder from a resource perspective. So there are much larger studies of antibody alone that have mostly seen this type of outcome. If anything, neutralizing antibody titers are more common, are higher in severe cases. And the general interpretation of that has been that over time, antigen drives those neutralizing, since the person has a bigger viral infection, antigen drives a higher neutralizing antibody response in the end. But in contrast, for virus-specific CD4s, we actually saw that presence of virus-specific CD4s was associated with reduced disease severity. And so that would be consistent with the CD4 T cells playing a protective role in controlling the viral infection. Or to turn it around, absence of a detectable virus-specific CD4 T cell response was associated with more likely having severe disease. Okay? And then a similar outcome with virus-specific CD8s. The presence of a virus-specific CD8 response was positively associated with less disease severity. Or again, absence of CD8s was associated with a higher likelihood of severe disease. And so with those, we then did two approaches to try and combine all of the data together. The first was a very simplified approach of saying, okay, we know there are three major branches of adaptive immune responses. What if we simply count those up for each individual? And so if a person has neutralizing antibodies, virus-specific CD4s, and virus-specific CD8s, that would be a score of 3. And if they have none of the 3, that would be a score of 0. And what does that look like? And that indicated that adaptive immunity was associated with better control of the virus. And in fact, it was absence of a substantial adaptive immune response was associated with more severe disease outcomes, consistent with the adaptive immune system playing protective roles in this disease. We had one anecdotal example of a subject who was negative for neutralizing antibodies. Other studies have found, made similar observations. And interestingly, in this individual, even without detectable neutralizing antibodies, this person had a mild course of disease, and they had a robust T cell response, both a CD4 and CD8 T cell response. Again, consistent with this statistical association between T cell responses and reduced disease severity. So that was the simplified approach, and then this was the opposite approach, where as an exploratory analysis, we took all of the data we'd collected, every antigen-specific parameter, as well as some more general parameters, and we asked, by looking at every possible pairwise comparison for the entire group in the study or the acute individuals, we did both, what patterns are there? And so this was a Spearman rank order, two-tailed statistical correlation, visualized as a correlogram, if you haven't seen these before, and it was a hierarchical clustering, okay? And there's a lot of data here, but to us, the big patterns and take-home messages were as following: that it looked like coordinated adaptive immunity is protective immunity. That the CD4, sorry, I should clarify. Positive correlations are in blue, negative correlations are in red. And in most individuals, we saw strong correlations between actually essentially all of the virus-specific CD4 T cell parameters, and strong correlations between the CD4 T cell response and the antibody response, and then down here, the CD4 T cell response and the CD8 response. And in fact, if we look through all of those individual pairwise comparisons for which of those are most strongly associated with disease severity, what we see is indeed that the total virus-specific CD8 response, virus-specific CD4 response, or actually the TFH response, were the strongest associations with lower disease severity, i.e. High CD8, CD4, and TFH responses were associated with lower disease severity, and absence of such was associated with higher disease severity. Additionally, we also observed an age association. And so it's well recognized that age is a major COVID-19 risk factor, probably the biggest COVID-19 risk factor. But that doesn't necessarily have anything to do with immune responses, and even amongst immune responses, adaptive immune responses. But when we take a look at our data, if we split the group into under the 65 and over 65, what we observe is that in the under 65, there are, again, a lot of strong correlations between the different components of adaptive immunity. But over here, a lot of that's lost. So here, all of these blue boxes between the CD4s and antibodies are lost, and generally the same thing between the CD4s and CD8s. And what we concluded from that was that was consistent with poorer T cell responses in older individuals. And we explored the data to see if there were indications as to why that might be the case. And a relatively simple explanation presented itself as one possibility, which is that naive T cell frequencies drop substantially in people over time. And we see that in our cohort. We see that both for people who have COVID-19 and unexposed. And this general observation has been seen for 2 decades now, that as people age, they have fewer and fewer, they have a smaller and smaller naive T cell repertoire. And so if you're faced with a new viral infection, a novel virus, much of your T cell response is expected to come from the naive T cell repertoire. And so it stands to reason that if people have a smaller naive T cell repertoire to select from, that their T cell response may be slower or weaker than desirable. And so that may be a relatively simple risk factor for COVID-19 disease severity. And indeed, this is data from Donna Farber's lab, directly looking at human T cell output as a function of age. This is a paper from them from a couple of years ago, and that up to about age 40 or so, thymic production is quite high, and then it drops dramatically. And yeah, the data we're showing are consistent with that. And actually, I just had my birthday, so I think I'm going from this data point to this data point in this particular graph. And so that's not, to be clear, right? That's not proof of what's going on, but it's a relatively straightforward association that seems like a reasonable possibility. And so to summarize that study, we measured virus-specific CD8s, CD4s, and antibodies in the same people in acute disease and convalescent. And the overall conclusion from the study was that it looks like coordinated adaptive immunity is protective immunity, that all 3 of these branches of the adaptive immune system could be working together. And that's really a fairly common phenomenon in viral immunology, that that you have these different components of the immune system for a reason. And that it, from the data we had, it looks like T cells do the heavy lifting, which again is a relatively common phenomenon. When you've been infected, antibodies are less functional, and you can frequently have a need for T cell responses to clear the infection. That's consistent with observations seen in the human population with A-gamma globulin-anemic individuals, or people who don't have B cells and therefore can't make antibody responses or extremely weak antibody responses, as well as people who are having pharmacological depletion of B cells with rituximab. Those individuals are generally doing okay with COVID-19 infection, suggesting a major role for T cells in the context of the infection. And as I described, age is associated with those differences, and possibly because the older you get, the harder it gets to make a good T cell response to, or a fast T cell response to, a new viral infection. Not all the data in the literature is consistent or agrees, but we do think a lot of the data in the literature is consistent with these outcomes. And part of differences in the literature can be attributed to timing of sampling. So with that information as background, here's our working model of how we think the relationships are between immune responses and COVID-19 disease severity. This is one working model. This is not definitive, but I think it provides a helpful framework for thinking about the infection and for designing experiments, as well as for thinking about vaccines and therapeutics. This was published this past month in Cell as part of a review article, and we actually developed this back in October, November. So in a generic viral infection, right, what should happen is you have an innate immune response that occurs very quickly, usually starting within a matter of hours, followed by an adaptive immune response substantially later, let's say a week later. In an average SARS-2 infection, usually a virus has to have at least one immune evasion mechanism to be able to cause disease in humans, okay? And it's frequently important to understand that immune evasion mechanism to understand the pathogenesis of the disease. It doesn't have to be just one, but usually there are at least one. And in the case of SARS-2, I think it's been very clear by a lot of labs that it's very good at evading early innate immune responses. And I think early innate immune evasion, particularly of type 1, type 3 interferon responses, really may be the one big trick of this virus, and that much of COVID-19 can really be explained as downstream consequences of that event. Because by having this early, SARS-2 is particularly stealthy at avoiding triggering an early innate type 1, type 3 interferon response. And as a result of that lag in the innate immune response occurring, the virus gets a head start on its replication, okay? But additionally, adaptive immune responses largely depend on innate immune responses. So those initial alarm bells of the type 1, type 3 interferons going off are usually a prerequisite for an adaptive immune response. And so a delay in the innate immune recognition of the virus likely also results in a delay of the T cell and antibody response to the virus. But in an average SARS-2 infection, that T cell and antibody response occurs fast enough to control the viral load within a reasonable amount of time, frequently fast enough so that it's actually an asymptomatic infection, right? So then, and this is just the same thing, just made vertically, we consider severe SARS-2 infections and ARDS to be a continuation of that same model. That what could be occurring is that instead of an average delay in the timing of the innate immune response, if that delay is even later, now you end up with serious problems because the virus gets too much of a head start and you get too much of a delay in a T cell and antibody response. And I think there's compelling evidence of this in humans from multiple studies, from in vitro and in vivo work from say the Tenover lab, but also really the Casanova science paper showing that people with type 1 interferon deficiencies or defects are at extremely higher risk for having severe fatal SARS-2 infections, okay? And what could be happening in these scenarios is that the innate immune response to the virus is too late, and so there's a substantial delay in the T cell response. This is consistent with our data where we saw, right, people who were hospitalized and who ended up with the greatest disease severity were people who were actually failing to make T cell responses for 15 days, 22 days post-symptom onset. That's a really long time to be failing to make a successful T cell response. And then there's a beautiful recent study by the Bertoletti lab looking at the kinetics of T cell responses over time. And indeed, people who made rapid T cell responses to the virus were very likely to rapidly control viral loads, whereas this was observed in severe cases in their study, and that there was no correlation between antibody responses and the kinetics of the antibody response and whether somebody had an average or severe case. It was really heavily dependent on the T cells. Again, consistent with our findings, and I'll come back to that. So if this response is too low and too slow, really the other piece of the puzzle here is that, again, in this simple model, what could be occurring is that in the absence of a successful T cell response or a fast enough T cell response, the innate immune system tries to fill that gap, tries to fill that vacuum left by the fact that the T cell response hasn't occurred yet, to try and bring the viral loads under control. And indeed, the innate immune system probably is successful at that for a period of time, but it's not sustainable. And at some point, you get too much immunopathogenesis from that innate immune inflammation, particularly from neutrophils and from myeloid cells, and severe disease occurs, okay? This is also consistent with most of the autopsy studies of COVID-19 have really reported, and lung biopsy studies of COVID-19 have really reported lungs full of neutrophils, which are cells of the innate immune system, and other myeloid cells, and really not that many T cells in severe disease compared to non-severe disease. I will clarify that everything about this model is about kinetics, right, and temporal changes. And so since T cell and antibody responses are exponential, kinetics matter. So at the onset or early time point of severe disease here, these might be low, and that can be explaining the scenario. But by the end of severe disease, right, the T cell and antibody responses may be high and equivalent, and that you've missed the time window for understanding cause and effect. So again, this isn't the only model that's available, but it can explain a lot of the data that exists. In the context of aging, right, this is pretty consistent with what's being observed in older versus younger individuals, and again, kinetics of responses. And from a vaccine perspective, this is generally, we generally consider this model a positive sign and an encouraging sign, because if the disease is behaving in this way, that was consistent with the possibility that vaccines could do quite well against this disease, because it's suggesting really absence of adaptive immune responses are really the problem here, right? Not adaptive immunity causing the disease. And the kinetics are the problem. The real problem with the disease here is that the T cell response is too slow. Well, the great thing about vaccines is that they're generating the immune response in advance, right? And antibodies, your best possible vaccines are always vaccines that have really high levels of neutralizing antibodies and have them for an extended period of time, because antibodies do their best job if they're present before you're infected. And so you can, in a normal infection, it's a race between the adaptive immune system and the virus. The vaccine gets rid of that race. And so having the antibodies at time zero means you're much more likely to be able to be protected. And then this model also suggests that if the virus gets past the antibodies, T cells are likely to contribute to facilitating control of the virus, at least to prevent severe disease. Okay. Lastly, I'll say that I think all of this is very consistent with monoclonal antibody therapeutic trials in humans, which has been that they've generally not been successful in hospitalized patients, I think, which is consistent with it being too late. But then in an outpatient setting, if people get monoclonal antibody treatment very early, it can have an impact, and definitely a clinically relevant impact on preventing hospitalization. So I am very much on team monoclonal antibody for very early treatment in an outpatient setting. But those data, actually, particularly from the Regeneron clinical trial, are actually very consistent with this model, and that a model of T cells doing a lot of the heavy lifting during an acute infection, because even with a massive infusion of monoclonal antibodies, so that people had neutralizing antibody titers of like 1 to 100,000, the viral loads over 7 days only dropped a few fold. Okay, whereas in contrast, in that same clinical trial, if people seroconverted on their own, i.e., we're probably making T cell and antibody responses on their own, their viral loads dropped 1,000-fold, not a couple-fold. So lots more data to collect, and there are definitely more than one model that can be considered here. And I don't think that any single model can explain all cases of COVID-19, but here's our model and how it fits with the available data, okay? So with all of that in mind, really the next big question for us was, what about immunological memory to this virus? How much is there, and how long does it last? And given what I just said about potential roles for antibodies, CD4 T cells and CD8 T cells in control of SARS-CoV-2 infections or limitation of COVID-19 disease, as we've done for all of our studies the past year, we felt it was important to measure all of the major branches of adaptive immunity in each individual. And in the context of memory, that means memory CD4s, memory CD8s, memory antibodies, long-term antibody responses, but also memory B cells. Memory B cells are B cells that are virus specific, but they are not actively secreting antibody, but they could respond to a recurrent viral infection. So for this study, Alex Setty's lab, Professor Daniela Weisskopf, and myself gathered together a cohort of 188 people, representing a good deal of diversity and a range of disease severities, and measured all of these responses in all of those individuals, okay? And this is almost certainly the largest such study that's ever been done for any viral infection. And importantly, from a number of our studies on immunological memory over the years, we've concluded that really you have to have data out to 6 months post-infection, at least 6 months post-infection or vaccination, to have a good sense of the kinetics of immunological memory in people. Unfortunately, there are not good predictors at early time point of the durability of immune memory. And so you really need about 6 months of data to get a sense of both the magnitude and kinetics. And so that's what we did here. And in fact, in the end, it ended up being a study of 8 months. And so here we've got 188 people. On the left side, these are the cross-sectional studies, cross-sectional data sets, pardon me, cross-sectional data of people out to about 8 months post-symptom onset. And all of this was recently published in Science this past month, with the preprint originally in November. So on the left side, we've got this slide is about the antibody responses, and the left slide is the cross-sectional data. Here we've got spike, RBD, and neutralizing antibody titers. These were done in Erica Safire's lab here at LJI. And on the right side, we had a subset of the people in the trial, in the study, who we had longitudinal data for. We had at least 2 different samples from, and this was about 40 to 50 people. And so this was a sanity check for us to see whether the cross-sectional interpretations were meaningful. I've got some very excited jet planes in the background at the moment, which in all cases they did. And so the cross-sectional data was actually reasonable. And in general, spike in RBD and neutralizing antibody titers were retained substantially over this window of time, which is consistent with several much larger serological studies that are available, right? As I said, it's more straightforward to do serological studies, so there are some much larger serological studies, and our data, I think, connect well with those. And here I'm thinking of the very large study out of Mount Sinai by Florian Kramer and colleagues, as well as the large study in Iceland, both of which observed 90% plus of people being positive for antibodies out at 6+ months post-infection. We also saw IgA being maintained, which is a valuable contribution to the literature, and I think reasonably consistent with what was shown by Jennifer Gomerman and colleagues at University of Toronto, which I'll come back to. So memory B cells, we looked at antibodies, then we looked at memory B cells. We did this by flow cytometry, looking for B cells that could bind to SARS-2 spike, as well as amongst those, the cells that could bind to RBD. RBD being important because this is the domain of spike that most, probably 90% of neutralizing antibodies are specific for. And this work was done by scientist Alex Cato in my group, and really we felt like this was quite beautiful data. And in the end, this is probably the most surprising finding from the study, which was that memory B cells, people had more memory B cells at 6 months post-infection than they did at 1 month post-infection. And that was true for spike-specific memory B cells, RBD-specific memory B cells, as well as nucleocapsid specific memory B cells as an independent antigen. And it was also true for the longitudinal data from per individual. And it was also true if we specifically looked at the IgG-specific, spike-specific memory B cells, that there were more at 6 months than 1 month. This is consistent with an earlier finding by Miriam Pepper's lab, where she observed more at 90 days than 30 days. And it's also consistent with a paper just published by Michelle Nussensweig's lab, looking at specifically a 6 month time point compared to a 1 month time point, and they saw the same type of increase. So it's consistent in the literature. It's also, interestingly, consistent between infections. There's not much other data available, but Laura Walker's group looked at, I think, 4 individuals with, maybe 6, with yellow fever virus vaccine. And while that was just sort of a study on its own at the time, they actually observed a relatively similar pattern for that vaccine as what's being observed for SARS-2. Interestingly, a rather small percentage of these were IgA, even though circulating IgA titers were significant. And IgM memory does exist, and it's quite interesting memory. But what we observed was that it actually drops pretty rapidly over time and appears to be lost with time. Okay. Postdoc Jose Mateus and Alex Fede's group measured memory T cells in these people, memory CD8s in these people, and they did this in an exhaustive way. They actually, to be very thorough, measured the CD8 T cell responses to every single one of the SARS-2 proteins. And here's that data combined for every single individual into a single graph. For which we observe about 70% of people were positive for memory CD8s at 1 month post-infection, and that's consistent with our previous study and consistent with some other reports in the literature. And it went to about 50% of people being positive at 6 to 8 months post-infection. The half-life of this response that we inferred was approximately 4 months. There's not much data on this in the literature, but the best available data is really from Rafi Ahmed's lab, looking at the yellow fever vaccine and tetramer-specific cells, and they actually observed a similar half-life to the yellow fever vaccine as we observe here, indicating that these are quality memory CD8 T cells after SARS-2, and similar findings longitudinally. Of note, a high proportion of these had a TAMRA phenotype, which is uncommon after an acute viral infection, but that's what we observed. Next, memory CD4 T cells, similar experimental approach. And here we see almost everybody had detectable memory CD4 T cells to the virus and really quite high levels of those. And still, I think 90% of people were positive for memory CD4s or more than that out at 6 to 8 months, with a half-life a little bit faster than the CD8s. We also specifically looked at spike-specific CD4s because spike is what's most important for neutralizing antibodies, and the CD4s provide help to spike. And again, similar responses in kinetics there. Finally, we looked at the TFH cells, and the TFH cells were present and they were maintained well over the 6-month window of time. Interestingly, a significant fraction of them were high for CCR6 over time, which is possibly an indication of mucosal homing of these memory cells. So if we take all of that data together, what do we make of it? Well, this is really the first such study that's had these kind of data, and so it's given us some interesting opportunities to look at relationships between memory compartments. And really a couple of the main conclusions are that immune memory is complex. These different components of immunological memory do not all follow the same kinetics, and that there's a lot of heterogeneity between people, and that seems to be a function of this initial SARS-2 infection, resulting in 200-fold ranges between people. Practically speaking, we wanted to make some sort of composite metric to try and understand this immunological memory in people. And so, as noted before, we don't know direct mechanisms of protection in people against COVID-19. And it may be years still before we fully understand different parts of immunological memory that can provide protection. But what we felt was that it was reasonable to provide a first estimate of that by looking at, of all the metrics we measured, which were ones that were most likely to be contributing to protective immunity in some way. So one is RBD IgG, because that's the majority of the neutralizing antibodies, and then RBD specific memory B cells, because again, RBD is the target of most neutralizing antibodies, then total virus-specific CD4s, total virus-specific CD8s, as well as spike IgA. And if we take those 5 components and ask, how does that look across a population of people? At 1 to 2 months post-infection, most people had a lot of those. And out at 6 months post-infection, what we observed was that about 95% of people, actually, and we can just jump to this, showing it as little people, about 95% of people were positive for 3 out of 5 of these. We consider that a positive sign, in general, good news. and that's probably sufficient immunological memory to protect against the virus, at least at the level of preventing hospitalization level disease, probably for multiple years, given the kinetics and the magnitudes that we observed. That's a best guesstimate, right? But that's what we were inferring. That does not mean this would be enough to prevent people from getting infected at all. That really depends on how much neutralizing antibody is around and how hard sterilizing immunity is. But to limit disease or prevent hospitalization, these components of immunological memory look like they should probably suffice. But notably, a small fraction of people, right, about 5% of people, really had quite limited memory. And so we wonder if those people are more susceptible to reinfection or more severe disease. We did see some components of heterogeneity. Hospitalization contributed some, gender contributed some, but not enough to explain it. And so there's a significant amount of heterogeneity in memory that's not explained by the factors we know. And lastly, we looked at the relationships between these different components of memory, again, because this was a rare opportunity to do so. And interestingly, these different compartments of immunological memory have different relationships with each other. Probably most notable from a practical standpoint, that you would love to be able to do a serodiagnostic test and have that be predictive of T-cell memory. And our data indicate that that's not the case, that there's poor predictive power of RBD IgG for the magnitude of memory CD4 T cells in people, unfortunately. So here's a summary of all of that, which I just talked you through. This did get a lot of attention, and there have been multiple studies coming out since that have generally been consistent with this, both on the T-cell memory side, the antibody side, and the memory B-cell side. And protective immunity itself has also been directly measured in several other studies that didn't track immunological memory, but tracked just did people get infected and did people get sick. And I think most notably a New England Journal of Medicine study out of the UK that tracked 1,200 people over 6 months and observed that reinfections were rare and repeat sickness was quite rare. Okay, so I'll stop there and thank people, and then be happy to take questions. As I said, all of this was done in collaboration with Professor Alex Setti. That recent science paper on immunological memory was co-led by Professor Daniela Weisskopf, who did a fantastic job with the logistics of getting all of those samples. The initial paper of average cases of COVID-19 was led by a very talented postdoc, Albo Grifoni. The memory B-cell data done by Alex Cato. I mentioned Jose Mateus. Jennifer Dan is an assistant professor at UCSD and was responsible for all of the serology. Sydney Ramirez, infectious disease fellow in my lab, did a fantastic job recruiting people for the acute study, particularly in hospitals. And Carolyn Motorbacher led that overall study in the immunology thereof. So I'll stop there and be happy to take questions. I'll turn it back to you.
Thank you, Shane. That was a great talk. And clearly we've all been enjoying the talk because we have a lot of questions. So let's get started right away. I'm going to get started with the question about the immune memory. So we've been hearing about, we heard about immune memory, and given the fact that we tend to remember, our immune system tends to remember, what are your thoughts about changes to the vaccine regime, especially the second dose, the timing of the second dose? Because we've been hearing a lot of debate about spacing the second dose out or even giving half the dose, right? So given this memory, that this memory exists and is still robust for a good amount of time, do you think this would be a good approach to space the second dose out? Or do you think we would be paving way for more fit escape variants because the B117 was isolated from an immune-compromised individual? So yeah, what are your thoughts? We would like to hear your thoughts on this.
Yeah, great question and series of questions. So, so B117 probably, highly likely was it evolved in an immunocompromised individual. There's no direct history of where that came from, but there are multiple published case reports of similar types of individuals with lots of mutations. So I definitely think that's an appropriate conclusion. In terms of immunological memory to the vaccines, unfortunately, immunological memory is complicated enough that to understand memory to the vaccines, you have to directly measure memory to the vaccines. So the memory that we measured to COVID-19 cases is a helpful reference point for the vaccines to show that, okay, immunological memory in each of these components can happen to this virus and can happen reasonably well. But whether individual vaccines do that and how well they do it does have to be directly assessed. Cross-referencing the immunological memory data to protective immunity data from these prospective studies, again, does suggest that it lasts a meaningful amount of time. The dosing questions about vaccines are very interesting. I think the quick answer is the phase 3 clinical trials for the RNA and Moderna vaccines had a specific timing of the immunizations. And those phase 3 clinical trials proved that those work, right? 95% protection against the virus. Those regimens were designed to try and develop protective immunity as quickly as possible, okay? Which would be that with a 2-dose regimen, trying to get that second immunization as quickly as possible, day 21 or day 28, to try and get full protective immunity. What the UK government did was to make a decision under emergency situation, right? It's not a decision they would have made otherwise, but they made a decision that they thought that they could save more lives by spreading out the immunizations and doing that for the AstraZeneca vaccine and the Pfizer vaccine. And that came under a lot of criticism, and I'm very much in line with the statement by the US FDA, which is that they did not support changing the schedule because there wasn't evidence showing that protective immunity would be durable after a single immunization if you stretched out for 3 months. But the UK government did actually have access to data that wasn't public about the AstraZeneca vaccine that's now public recently, that does look like after a single immunization with that vaccine, there is something like 75% protective efficacy out to at least 12 weeks. And so doing that vaccine on a 12-week regimen, I think is reasonably well supported by the data at this point. More broadly, immunologically, I've been saying whether you get your second immunization at 1 month, 2 month, or 3 month, the amount of memory and protective immunity that you'll have 6 months later after that immunization is probably the same, whether you get that immunization at 1 month, 2 month, or 3 month. If anything, it might be somewhat better if you wait a bit longer. Certainly papers by my lab and plenty of other labs have indicated that if you wait longer, there's a reasonable chance that your actually memory response is even stronger. The current regimens have been designed to get you protective immunity as fast as possible. So those are the factors at play.
Okay. Thank you. We will now move on to the audience question. We have a great question from Brian. Brian, will you please unmute yourself and ask your question?
Oh, great talk. Thanks so much. I have a question about your initial slides of I think 20 subjects that had recovered, and only 80% had an IgA response. And I wonder if you could comment on how that will, if that's at all relevant, it's not 100% on upper respiratory reinfection or staying contagious, these kind of questions.
Yeah, that's a great question. And thanks for bringing it up, because I do, so mucosal immunity is important, and I do generally try and bring it up in the context of this model as one of the things that we just don't have as much data about as we'd love to have. So essentially everything that my lab's looked at so far has been circulating immunity, circulating T cells, circulating B cells, and circulating antibodies. And that's because that's what's been more straightforward to measure. Certainly in terms of protection against a respiratory viral infection, you absolutely need immunity to occur at that site. and whether it be upper respiratory or in lungs. And what we've seen in the literature is that circulating antibody titers have been reasonably reflective of antibody titers in saliva, okay? And so that's work by Jennifer Gomerman and others. And so we think the antibody measurements in the blood are reasonably reflective of what's going to happen at those mucosal surfaces, but it would be really valuable to have more direct evidence of that. It's quite hard to do those measurements in nasopharyngeal spaces. It's just technically very challenging. So a lot of people are using saliva as a surrogate. On the T cell side, actually, the same types of questions come into play. Are there, where is it? And I do cover this in the review that was just published, that yes, circulating responses matter, but in the end, those responses have to be recruited to the actual sites of infection. And so are they there, and are they there to what degree, and do they stay there, are important questions that remain to be resolved. In terms of the lung, at least, there are published pieces of data on it that these T cells in the context of infection can go to the lung, and that if anything, there are more T cells in the lungs of people with less severe disease than with more severe disease. Again, consistent with, if you are making a good T cell response and it is going to the tissue that's infected and going there fast enough, that's a positive thing for controlling the infection. How much IgA is present in mucosal surfaces and how long it stays there remains an unknown. A classic vaccine situation is that it's much easier to induce long-lasting IgA than IgG. And so a common vaccine goal is maximize the amounts of IgG, and that compensates for any absence of IgA, because IgG will get secreted at mucosal surfaces, even if it's less efficient than IgA. And really, that's true for a number of vaccines, but I'd say the poster child for that, really the most dramatic example, is the human papillomavirus vaccine, the HPV vaccine, which is, it's a shot in the shoulder. It just elicits IgG, no IgA at all. And it has to protect against a mucosal pathogen, HPV, that only infects the outer epithelial layer of the vaginal mucosa. That is a really high bar for protective immunity. And nevertheless, it's a vaccine with something like 99 or 99.9% protective efficacy. So that's the mentality, that it's fantastic to get IgA, and you definitely want it at the mucosal surfaces to the extent that it can be accomplished. But practically speaking, IgG can get the job done and can be easier to elicit in many contexts.
Thanks so much.
Thanks, Shane. Thanks, Brian. The next question, we have a question on immune-compromised population from Meghna. Meghna, would you please ask your question?
Yeah, thank you, Dr. Crotty, for a great talk. This was super interesting. I was wondering, and I had questions about, we know that cancer patients are immunocompromised, but do we know enough about how the vaccines might work in cancer patients, or how does the immune response look like, or the generation of antibodies would look like in these subgroup of patients?
Yeah, it's a really good question. And I'd say there were initially some confusing and conflicting recommendations, which is not uncommon, in a rapidly changing situation. But that the CDC recommendation has been pretty consistent, that the authorized vaccines in the U.S., the Moderna and the Pfizer vaccine, are recommended for immunocompromised individuals, and that essentially the expectation there is that those vaccines are completely safe in immunocompromised individuals, because they're not live vaccines. Normally, the only recommendation against vaccinations in immunocompromised people is if it's a live viral vaccine that they may not be able to control. But there's nothing alive about the RNA vaccines, and so there's no risk there, there's no safety concern there. And that the expectation is that the vaccines are likely to be effective in immunocompromised people. Now, the phrase immunocompromised covers a wide range of people, right? And it depends, even amongst the subset of those that are cancer patients can be on a wide range of either immunosuppressive drugs or intrinsically immunocompromised for various reasons. So there's no single answer there. And to some extent, it comes down to this more holistic model, right? Of all the different parts of adaptive immunity. And at the moment, it seems plausible that antibodies, CD4s and CD8s can all contribute to protective immunity, at least to some degree. And so if somebody's immunocompromised in one of those components, it's still plausible that vaccines could generate some meaningful protective immunity, which would be better than no vaccine at all. So that's the status.
Okay, thank you.
Thank you. Our next question is again another audience question. Jessica, please unmute yourself and ask your question.
Hi, thank you so much for this excellent talk. I think you answered one of my questions already about the role of IgA, but I was just curious if you have any thoughts on the potential success of a vaccine that elicits robust SARS-CoV-2 specific T cell responses, but not neutralizing IgG.
Yeah, that's a really good question. So, I'm obsessed with vaccines. I've pretty much studied vaccines for my entire professional career. And I love antibodies, CD4 T cells, and CD8 T cells. For a vaccine, it's a pretty simple rule of thumb that the best vaccine, your best situation with a vaccine is to elicit really high titers of neutralizing antibodies and sustain those neutralizing antibody titers forever. Like that's your best case scenario for a virus that's susceptible to neutralizing antibodies. And so, for example, SARS-CoV-2 definitely is susceptible to neutralizing antibodies. And so that is your best case scenario. Now, there you get the question of, well, how much neutralizing antibody do you need, right, to be fully protective? And then can you actually sustain those forever? And so that's when you then get into, or would it be protective against all possible strains, right, all possible variants? And so that's where you start getting into questions of, do T cells also contribute? So if the virus gets past that initial antibody, is it helpful to have other components of the immune system? And those can be dependent on a number of factors, a number of which I described over the course of this talk, but one that I didn't highlight enough that directly relates to your question is the issue of timing and speed of disease onset. So if you are faced with a very rapid disease, and bacterial meningitis is a good example of this, that if you get infected and it gets into your spinal cord quickly, you can be dead in 48 hours. That's a very narrow window of time for your immune system to do something protective, okay? And in that type of scenario, it's generally almost all about the antibodies, because the antibodies, it's basically, did you have enough antibody to keep the infection from happening at all? And that's your plus minus. But if there's a bigger window of time, there are more opportunities for the memory CD4s, memory CD8s, and memory B cells to contribute to control. And one of the compelling things about COVID-19 is that, severe cases, fatalities are on average happening like 20 days after symptom onset. So that's probably 25 days after infection. Okay, that's a long time for your immune system to have the opportunity to do something helpful to control that disease before it gets too far. And since T cell responses are exponential processes, even if you don't have enough T cells at time zero, if you had some memory T cells here, right, that they would, actually I have a slide on this that we made for other reasons. Where is it? There we go, it's here. Here would be, so in green would be virus, or and green is disease and yellow is virus. Okay, so maybe you'd normally have this, but if you actually had T cells in advance, maybe they could come up and not completely prevent you from having any infection or disease, but they could do enough to limit the disease severity. And they also might accelerate another wave of antibody response, which again would do the same sort of thing. Now, your best case scenario with a T cell vaccine is if you can get tissue resident memory T cells. So T cells that would actually be in the nose at the site of infection. And in that scenario, there's good experimental data in mouse models from people like David Baltimore and others, that if you can have TRM at the site of infection, they can really wipe out the infection almost immediately. So if you could have a vaccine that could do that, that would be a fantastic outcome. And there's a Nature paper from Dan Baruch that indicates, I think, that in monkeys who've been previously infected, they do have T cells. It wasn't directly shown that they were in the nose, but they did have T cells that could blunt the infection very rapidly. So in a classic T cell vaccine, right, where if you wouldn't have tissue resident memory, it's unlikely that would be as good as antibody because the kinetics would be too slow. But if you could really get tissue resident memory cells at the site of infection, yes, a T cell vaccine would have a chance.
Thanks so much. I appreciate your response.
I know it was kind of a long response, but
no, it was great. Thank you. I appreciate it.
Very clear.
So much detail in this. I'm learning a ton. Keep doing what you're doing.
All right. We
10:15. Should we do a final one?
Okay. One last one. Patrick has a great question on escape mutants. Patrick, go ahead.
Yes. So I was wondering, in the context of evading the immune system and acquiring escape mutations, how common is it for viruses to acquire a major advantage in order to evade the immune system without first going through a species jump?
I'm not sure where the species jump part comes in, but okay. I mean, how common? I think the species jump part is important. I should have said about this slide. This was a backup slide. This is from a Nature Reviews Immunology article from a couple months ago. If anybody wants to reference, anybody wants to go find it, and where it describes these models in detail. So, I mean, obviously the several of the current variants are of substantial concern. I think that the answer I can give is all viruses mutate, and the big question is, do they mutate in ways that they can escape immunity in a substantial way, right? And that's going to depend on the kind of virus it is, and I think it also depends on whether that virus has had a recent species jump. And I think it's definitely more likely to occur for a virus that's had a recent species jump, because they're just it's more of a punctuated equilibrium kind of evolutionary situation where there are lots of different mutations that could occur that would improve the viral fitness for the new host. And I think what was a bit surprising was that we didn't see much of that through a lot of 2020, and then we saw a lot of it right at the end of 2020. To try and answer the question, I think more directly, one of the things that immunologists have looked for in the immunological data, well, for one, was for us, was the T cell response narrow or broad, right? Which I talked about in the beginning. Because if it's a narrow T cell response, that's concerning, because then if the T cells are playing a role in control, well, the virus might be able to escape that because there's not much that the virus has to escape. But instead, what we saw was the T cell response is quite broad, with probably a dozen CD4 epitopes and a dozen CD epitopes per person, and those epitopes being different from person to person. That is a very hard situation for a virus to evolve around. There's just too much diversity there. So it's unlikely that variants would be able to evolve to escape T cell immunity in COVID-19 cases. On the antibody side, the same type of concepts have been in play. People have been looking at those antibody responses and asking, do all the antibodies bind to just one site, or do they bind to a number of sites? And how diverse are those antibody responses? And the answers, really last summer from people like Michelle Nussensweig's lab, Pam Bjorkman's lab, Dennis Burton's lab, and a number of teams elsewhere have been that the antibody responses are quite broad, that there's not just one antibody response, there's diversity. And so that makes it harder for the virus to escape. Unfortunately, it looks like what has happened, at least for the South Africa variant, is that it has managed to escape several classes of antibody responses at the same time. And I think evidence from the Nussensweig lab, David Ho's lab, and Penny Moore's lab is all pretty compelling there. And now the questions become, is that a one-time thing? Or can the virus do that over and over? And that's an unknown. What I've said and still think is that the virus still has to recognize its receptor. There's a limited number of mutations the virus can make in the spike protein and still recognize the ACE2 receptor well and deal with this polyclonal antibody response. And so the fact that the antibody response is polyclonal and that it's a conventional receptor interaction, I think there are a limited number of ways that the virus can escape antibody responses. But it's certainly had plenty of surprises in the past 2 months. So we are going to have to get the data to see.
Thank you.
Yeah. Well, Shane, this is amazing. So much data. Thanks for being so generous with your time and answering questions so carefully. I know we said that was the last one. Would you indulge me a very quick blue sky question before we sign off?
From you?
From me.
Oh yeah, sure.
Okay, so
host prerogative, right? You get to ask at least one question.
Thank you, very, very generous. So you walked us through how we're losing our naive T cell population as we age, and we can get in trouble by that. Do you foresee maybe 10 years in the future? Do you think that we could be getting infusions of stem cell derived naive T cell populations to people of a certain age, such as we are, to kind of restart the clock?
Yeah, it's a good question. Because one of the things I didn't highlight about it, but you could see from it, there's a diversity between people, right? Some people were really down in single digits of naive T cells, and others had reasonable populations. There are a couple of aspects of that. Okay, so one is, well, can you just expand these populations? And the notion would be you could, and you might then have more of those cells, but it'd be the same repertoire. And so if the diversity of recognition events, if just the total number is a problem and they're just too rare, you could potentially bump that up with some treatments, treatments that get naive T cells to proliferate. But still, essentially the library of T cells would be the same, right? And if there are holes in that repertoire, you wouldn't be filling the holes. And we don't know which is the bigger issue. And so I do think probably the most relevant paper on the topic is this one by Donna Farber, and looking at the thymus, because the thymus is the source of production of the new T cells, and it drops dramatically. So, to me, the question there is, it looks like there's still a little bit of activity here, and this would be where the new specificities would be coming from, whatever that is. And so could you re-energize that organ to some degree? I think that's the question, and I definitely think it's worth exploring.
Okay.
And there's plenty of time because I'm not going to be way out here for a while yet. So.
Well, I'm a little further down that curve than you, so it's a little more urgent to me.
Fair enough. Well, yeah, thanks for having me. Great questions. And I'm glad not everybody was just staring at a photo of Tony Fauci for an entire hour.
Although I think that would have just boosted our spirits so much just to look at Tony for an hour.
All right. Thank you all for your time.
Okay. Thanks, Shane. All right. Keep it up, man.
Bye.
Bye. Everyone catch this talk next week. Galit Altair. And stay safe.